Hello friend, welcome to Tanya's Biology. Today I will discuss about the generation and conduction of nerve impulses. In my previous video, I have already described about the structure of neuron, where the myelins are present, where the non-myelinated portion are present, how the information are showing the saltatory conduction. These are the terms that I have used already in my previous video. If you want to get the video, please subscribe my channel. Okay. In this video, I will discuss about the conduction procedure of nerve impulses. Okay. What is nerve impulse? How it proceeds? Each neuron receive an impulse. Each neuron receive an impulse from and makes pass it to the next neuron and make sure the correct impulse on its path. Okay. That means there are two portions. One is presynaptic knob, another one is postsynaptic knob. If this is a neuron and this portion is known as the axon terminal. This portion is known as axon terminal. Where the axon terminal is present, the next neuron will start from this region. Okay, the next neuron will start from this region. That means information need to be carried from this portion to this portion. Presynaptic knob to the postsynaptic region, pre to post. Okay, that means the information need to be corrected. The correct information need to pass. Okay, how this impulse are providing information, how this impulse coming from that region and move to that region. In this procedure is divided into 6 steps. Nerve impulse generated and conducted in 6 steps. What is the first step? It is a polarized state. The first step is known as polarized state. What is the polarized state? In this polarized condition, sodium and potassium channels are present in the membrane. Sodium and potassium channels are present in the membrane. If you see, this is the picture. This is a polarized state. Polarized state. In this polarized condition, there is a channel. There are 2 types of channel or 3 types of channel. This channel is known as potassium channel. This channel is known as sodium channel. Okay, and this portion is known as sodium potassium pump. Okay, when we are talking about different types of ions, from where these ions are coming, why you are talking about ions? When the first animal present in the aquatic mode, if you say properly, in the aquatic region where the sodium chlorides are present, the first animal is evolved in the aquatic region or sea where the sodium chloride present. That means sodium ion concentration is higher in the outer medium. Whereas the concentration of sodium ion lower in the inner side. That means that ionic information are coming from that first animal which is evolved in many years ago. Okay, that means sodium concentration in outer region much higher as compared to the sodium concentration in the inner region. In case of nerves, the sodium concentration present in the outer side is very much higher present in the inner side, and that means sodium concentration higher in the outer side, but the concentration of potassium much higher in the inner side as compared to the outer side. But we need to know that there are sodiums are also present inside the cell, but concentration is low. Not that the sodium is totally absent inside the cell. No, that is not the phenomenon. Sodium concentration higher in outside the cell. That means sodium is also present inside the cell. Same as concentration of potassium is higher inside. That means the concentration of potassium is lowered outside, but they are also present in outside, but very little amount. Okay, but that from where the negativity comes from? This negativity comes from the protein. This negativity comes from proteins. Okay, proteins are present inside the cell. Proteins are present inside the cell. These proteins are negatively charged. These proteins are negatively charged. That causes a cell in the atmosphere of the cell inside is negative. Okay, from that idea, the negativity causes inside the cell. When an information come and it need to flow from one region to another region, the inside picture, the inside picture of the cell need to be positive. The inside picture of the cell need to be positive. Otherwise, the information will not able to go with the flow. Okay. In our blood, the concentration of the sodium and potassium need to be maintained. This if there are any problem in the electrolyte, then causing different types of physiological problem. Okay. Now we are coming to the polarized state. What is polarized state? Polarized state means when the membrane potential, or we can say the charge within the membrane, remain minus 70 millivolt. Minus 70 millivolt. In this condition, the polarized state are maintained. In this form, in this condition, some potassium channels are open. Some potassium channels are open, or through this open channel, the potassium are able to coming out. Not all channels. Make it very clear, not all channels are open. Some of the potassium channels are open, but all sodium channels are closed. All sodium channels are closed. That means potassium can move from inside to outside, but no sodium ion can able to penetrate from outside to inside. This is a polarized state. Polarized state. Okay, in this polarized state only the sodium channels are closed and some of the potassium channel will remain open. Okay, what about this pump? What about this pump? This pump, in the polarized condition, this pump help to maintain this charge. How this pump help to maintain this charge? Let me raise it. You can see here that there are the 3 receptor and outside of the membrane having 2 receptor. Okay, if some sodiums are present inside the membrane, please make it very clear. If some sodium ions are present inside the membrane, this pump will bind with the membrane. That means at a time 3 sodium molecules are bind, whereas if some potassium ions are present outside the membrane, the potassium ions are binding with that portion. After the binding, they just rotate itself. That means at a time 3 sodium ions and 2 potassium ions are binding. After the rotation, what will happen? 3 sodium ions will go out, 2 potassium ions will go in. Okay, that will happen. That means this pump helps to maintain minus 70 volt. Okay. That means what is the first step? It is a polarization of neuron membrane, where the sodium is on the outside and potassium is on the inside. Polarized means that electric charges on the other side of the membrane positive, while the electric charges of the inside of the membrane is being negative. Okay. The outside of the cell contain excess amount of sodium. The outside of the cell contain excess amount of sodium. We all already described that why the sodium is present outside is very higher. and the inside of the cell contain excess amount of potassium ion, excess amount of potassium ion. That doesn't mean that the inside of the cell doesn't have any sodium ion, they also have the sodium ions. Negatively charged protein and nucleic acid molecules are also inhibit the cell. This negative charge within the cell is created by the proteins that are present inside the cell and the nucleic acid molecules. Now, what is the second step? Resting potential. When neuron is inactive or polarized, it is said to be its resting potential. It means this way until a stimulus comes along. When it is present in polarized state, that means they are in resting phase where no sodium ion can influx and some of the potassium ions can go outside. These are the resting phase where the no information is coming. They are in resting mode. When information comes, what will happen? What will happen then? When the neuron is inactive, we already described there. What is the third step? When the sodium ions moves inside the membrane. When it will happen? When any kind of information that comes. When there is any kind of information that comes. The information comes in form of ion. The information is carried forward in form of ion. How we make it, please, if you focus that picture, you can see that how we measure this amount of minus 70 millivolt with the help of microelectrode. With the help of microelectrode. This microelectrode help to measure the condition of the, let me write it, to measure the amount of ionic concentration present within the cell, outside the cell. Okay, with the help of electrode we can measure it. If we say that we are hungry, the science wants to know how much you are hungry. They want to measure it. You are in pain. The science wants to know how much pain you have. They want to measure it. So that means science want to measure each and everything. So with the help of microelectrode, we can measure the resting membrane potential. That means this state is known as resting membrane potential, where there is no information. And cell are in resting mode. Okay. Let me change the slide. What is third step? When there is an information and neurons need to carry that information, what will they do? They certain change their configuration of the cell membranal channel. They certainly change the information. When the cell is present as 70 millivolt, minus 70 millivolt, when there is information, they change into minus 60 millivolt. They change the membrane potential into minus 60 millivolt. If I change the color, it makes it very visible. Okay, from minus 70 millivolt to minus 60 millivolt. When the membrane potential comes into minus 60 millivolt, certainly all the certainly all the sodium channel make open. Certainly a drastical change happen in the channel configuration. All the sodium channels open, and it will help to enter all the sodiums. When a stimulus reaches a resting neuron, a gated channel on the resting neuron membrane opens suddenly and allow the sodium that was on the outside of the membrane to go rushing into the inside. Okay, that I have already discussed you. That means they are talking about this phase. Okay, where there is no change in the potassium configuration, or there is no change in the potassium channels configuration. All the channels are open except some channels are all the channels are closed except some of the channels are open. But in case of sodium, all channels became open, which helps to influx the sodium ion, okay. The negative charge of the protein molecule, the neuron goes from being polarized to being depolarized. This state is known as depolarized state. Polarized state. Okay. Depolarized means when the channel goes from -70 to -60 to 30, -30, from -30 to 0 to 20 to 30, then 50. That means from minus to the journey from being plus. This state is known as polarized state. Okay, this state is known as polarized state. After more positive ions go charging inside the membrane, the inside became positive. This positivity comes from the sodium ion. As we know that all the sodium channels being open, sodium ion influx the cell from the outside. This causes the positivity within the cell. That means from polarized state, the cell undergoes depolarized state. That means all the potassium channels remain closed and some of the channels are open, whereas all the sodium channels became open. And that causes the influx of the sodium ion. What is the next phase? That means from polarized state we go further to the depolarized state. After the stimulus goes above the threshold level. What we are talking about the threshold level, I already discussed you. When there is a stimulus the membrane potential from 70 to become -60. This we are talking about this level, this threshold level. After reaching the threshold level, suddenly all channels are open. They thought that there is important information. They need to carry the information. They need to they understand the importance of the information. Then only they open all the channels. Otherwise, they remain closed, all the sodium channels. Okay, after the stimulus goes above the threshold level, more gated ion channels open and allow more sodium inside the cell. Okay, this causes complete depolarization of the neuron. And an action potential is created. What is action potential? That the journey from -7 to +70. The journey from -70 to +70 is the action potential when they reaches their peak. When all sodium channels are open, all, not only one or two, all the channels of the region remain open, and in sodium channels are passing from outside to inside. This condition is known as action potential. When they reach at the +50 level, or you can say the +30. This is a controversial part. They are from +730 or +50. These are the two measurement. Okay. That means polarized to the depolarized state, where we can find there is a action potential. Okay. What is the fourth step? After reaching the depolarized state, the highest portion of depolarized state, that means they are reaching the peak of the action potential, they need to repolarize it. That means they need to go to their old position. Okay, where the potassium ions moves outside and sodium ions stay inside the membrane. If you see that picture, you can see that now when the information need to carry forward, the polarized state need to get back. They need to go back to their polarized state from depolarized state. This process is known as repolarization. From depolarization, when they come back to the polarized state, known as repolarization, where the potassium channel became open and suddenly all the sodium channels became closed. Sodium channels remain closed and potassium channel became open. Okay, got it? That means when the depolarized state need to return to their old position, they need to repolarized. Repolarized. Then potassium channel became open and sodium channel became closed. Okay. So, potassium ions moves outside and sodium ions stay inside the membrane. After the inside of the cell becomes flooded with sodium ions and sodium gets closed and potassium gets open. With potassium moving to the outside, the membrane repolarization restored the electric balance. It is opposite of the initial polarized membrane. In this case, you can see that after the sodium ion entered inside the cell, then the sodium channel became closed. Then the sodium channel became closed. Okay, they are closed. And that moment all the potassium channel became open. That causes the concentration of the sodium inside the cell became higher and the concentration of potassium outside the cell became higher. That also causes negativity inside the cell and positivity outside the cell. But there is a difference. There is a difference. What is the difference? In the first case that I have saw that potassium in concentration inside the cell is higher, sodium concentration inside the cell is lower. That means sodium concentration outside the cell is higher. But after repolarized state, we can see that sodium concentration inside the cell became higher and potassium means sodium concentration inside the cell became higher, whereas the potassium concentration outside the cell became higher. That causes an ionic concentration difference. Now the pump show their activity. The pump with the help of ATP. They can't do it without the help of ATP. They are doing their activity with the help of ATP. Now they bind with the sodium ions that are present inside the cell. They bind with the sodium ion, 3 sodium ions inside the cell and 2 potassium ion outside the cell. And 2 potassium, let me change the color, 2 potassium ion outside the cell and just rotate. They frequently do their job. That causes the sodium concentration inside the cell became low and the potassium concentration inside the cell became higher. With the help of this sodium potassium pump, they utilize ATP to maintain their concentration gradient. Their ionic concentration within the cell and outside the cell. Okay, if we put all our information in a graphical pattern, we can see, let me erase it, that when this is minus 70 condition, they are resting potential. They are present in the resting potential. This phase is known as resting potential. After they get information, they change from minus 70 to minus 60. This is all about the threshold level. Okay. When the information went into the threshold level, all the sodium channels suddenly open and that causes depolarization. And that's how this action potential will form. Okay, when there is, we know that they reach in plus 50 millivolt, then they show the action potential. That means when the information reach, the whole scenario will change. When the information reach, the whole scenario will change. The ionic concentration became changed with the help of sodium ion and potassium ion. When the sodium channels are open, the concentration of the sodium inside the membrane increases, that causes from polarized state to the depolarized state, and they enter into the action potential state. After reaching that phase, they need to get back their polarized state. So they go under repolarization, and that causes from plus 70 to go through the minus 70 level. In this phase, potassium ion goes outward. They don't know how much they need to go. So somehow the concentration from minus 70 goes under minus 80 millivolt. This phase is known as hyperpolarization. Because these processes are going on process. They don't realize they need to stop. At minus 70 level. So they are going on and on and on. That causes hyperpolarization. That is the charge minus 80 millivolt. Okay. After they go into the hyperpolarized state, they need to go back to their polarized state from minus 80 to minus 70. At this phase, this is known as refractory period. This phase is known as refractory period. At that moment, if any other information come, except the first information, if any other information will come, they doesn't receive that information because there is already an information. Okay, when the first information is there, they don't receive any other information. They don't receive the second information. Okay, that means that we are talking refractory period. What is hyperpolarization? More potassium ions are on the other outside than there are sodium ions on the inside. When the potassium gets finally close, the neurons has slightly more potassium on the outside than it has sodium ion on the inside. This causes the membrane potential to drop slightly lower than the resting potential, and the membrane is said to be hyperpolarized. This period doesn't last long. What is the sixth step and the last step? Is the refractory period, where the potassium return inside and sodium return outside with the help of the pump. The pump utilize ATP to do their job. While the neuron is busy returning everything to normal, it doesn't respond to any other incoming stimuli. Okay. After the sodium potassium pump return the ion to their rightful side of the neuron's cell membrane, the neuron is back to its normal polarized state and staying in the resting potential until another impulse comes along. This is all about the information. In this process, information will pass from one region to the other region. Okay. In my next video, I will talk about the transmission of impulse through synaptic knob, how the neurotransmitter will work, what kind of neurotransmitter. There are two types of neurotransmitter. If you want to get my video, please subscribe my channel. Don't forget to like and comment.